Now for our realities of N of 1 and implications for the patient community panel, we'll be joined by Dr. Olivia Kim-McManus, board-certified child neurologist at the Rady Children's Hospital, and Dr. Kiran Musunuru, a practicing cardiologist and professor for translational research in the Perelman School of Medicine at the University of Pennsylvania. Before our speakers introduce themselves, I did want to highlight the poll responses if they've come in yet, letting our speakers know that we have quite a variety of individuals joining us today and what the top perspectives will be. All right, we're still not done with those. So what we're going to do is come back to that in a minute. We would encourage the audience to ask questions throughout the session today using the Q&A feature. Now I'm going to kick it over to Dr. Olivia Kim-McManus, followed by Dr. Kiran Musunuru, before we get in our panel questions to introduce themselves. So first, Olivia.
Kim-McManus. I'm Associate Professor of Neurosciences at UCSD and a pediatric neurologist, epileptologist running N of 1 trials for antisense oligos.
And I'm Kiran Musunuru. I'm a faculty member at the University of Pennsylvania, as you heard. I'm a cardiologist, I'm a geneticist, and I'm a gene editor, although recently I've crossed over into the pediatric realm.
Great. Thank you both for joining us. We really appreciate it. So just to start off, what exactly do we mean when we say N of 1? And what do we mean when we say personalized medicine? How are those similar and how are those different?
I guess I can start. So there's a lot of overlap, but I think formally speaking, N of 1 denotes a variant or a condition that is truly unique. You have a patient in front of you who has a genetic variant causing a disease that is relatively unique. Maybe it's in their family, but otherwise isn't represented anywhere else in the population of patients with that particular condition. Or it's a syndromic sort of condition that is extremely rare to the extent that the one patient in front of you might have a unique flavor of that particular syndrome. So in my mind, that's what I think of as N of 1. A personalized therapy in principle is any therapy for any patient that is tailored to them. It could apply to N of 1 cases. It could apply to N of few cases where there would still be a need for personalization. It's less likely to be relevant if you're talking about a more common rare disease, if that's not an oxymoron, where you have a condition like sickle cell disease, where everyone has the same genetic variant. And so there's more of a opportunity to make a single therapy that will serve the needs of a larger group of individuals, or maybe all the individuals with a particular disease. So, there's some, a lot of it is terminology more than anything else, but that's how I conceptualize it. Your mileage might vary.
No, and I agree with Dr. Musunuru that, an N of 1 and it's more specific beyond the personalized medicine because that can be tailored across the board, but and specifically more in terms of gene-based therapies, that there's nuances to a variant or a presentation that might entail a more individualized approach with similar platform approaches that are already in existence on commercial level.
Thank you. So the poll results actually are in, and it looks like our two largest categories are foundation and nonprofit leaders and caregivers or persons living with a rare disease. For those people, can you discuss what are some of the misunderstandings around N of 1? Both things that are deemed as positive and things that are true challenges that need to be addressed for all N of 1s. Kiran, would you like to go first?
Sure. So let me start with challenges, and then maybe we can get to misconceptions. So I think the challenge with N of 1 cases is that there typically is not a one-size-fits-all solution already readily available for them. There isn't a drug, there isn't a treatment, there isn't an intervention that is going to serve the needs of a patient who's truly an N of 1 case. And so perforce, if there is going to be an effort to help that individual patient there may need to be a high degree of personalization. Now, in my realm, I'm a gene editor, so I'm thinking about the use of CRISPR gene editing tools. I'm thinking about genetic changes that cause disease, misspellings in the DNA, if you will. So for me, in my area, and I don't want to extrapolate beyond that to all types of therapeutic approaches. I'm just talking about gene editing. But if there is a misspelling, then the concept would be that you create a version or a variant of CRISPR gene editing that will specifically correct that misspelling and make it the correct spelling. That's not to say there aren't other approaches. But again, if there are no existing therapies, I think that would be just as valid an approach to try to undertake as trying to generate an approach that will help a large number of people with disease. I think you have choices, right? I don't know if you have anything to add to that, Olivia, from your perspective.
No, I agree. And there's a lot of challenges with N of one, development of an N of one, whichever, whether we're talking about gene editing versus mRNA pathways, all of them have to be kind of uniquely tailored because of the nuance of an N of one. And so there's steps that come in beyond just identification of a gene variant. And those things come into play in terms of actual execution and delivery into an individual. And a lot of the other things that really have to come into consideration as you go in parallel, since it is individualized on a level of the genetic variant is also the tailored outcome assessments that are appropriate to an individual that might or might not be relevant for the broader population of that syndrome or that disorder, because it might be a little bit different, but tailored also in terms of clinical trial. Both in terms of the actual drug design and the protocol.
So we had a question come in that talks about this individualized medicine idea. And the question is, most research that's out there focuses on common mutations of different genes for a specific disease. And obviously, we need therapies for the less common mutations. How can people understand or evaluate when an N of one does make sense for certain unique mutations and when it might not make sense?
So I agree. There is certainly a bias towards more frequent mutations, more frequent variants that are found in more individuals, and a lot of that bias is reflected by commercial incentives, right? If we're talking about companies that are in the business of developing drugs, and it takes the same amount of effort to make a drug that will help, say, 10,000 patients versus one patient, then they're going to go with the math. They're going to go where the commercial opportunities, where they can get the most return on investment. And for better or for worse, that's capitalism at work, right? That's the purpose of a company, is to make at least a return on investment, and then ideally, if it has investors and has a board and whatnot, I'm thinking about profitability. Academics, I would argue, also have something of an incentive in that direction as well. If all else being equal, if you are working, to make a new therapeutic approach for a disease like sickle cell disease, that's going to tend to get a lot more attention in terms of where you can publish in a more high-profile journal, in terms of getting attention for your work, in terms of getting funding for your work, than an N of one case for a disease that no one has ever heard of. That's not always true. Our own recent efforts focused on an N of one case with a disease probably no one has heard of, CPS1 deficiency, and it ended up getting a lot of attention. So I don't want to paint with too broad a brush. But generally, it's not just industry that has these incentives. It's also academics who have these incentives. And there's even, sort of a sense that if you can only put in so much effort because you're time limited and resource limited, you want to do the greatest good for the greatest number of people. I certainly understand that sort of utilitarian impulse as well. But my personal take on this is you want to resist that impulse as best as you can. And so when my scientific collaborator, Rebecca Ahrens-Nicholas, who I've been working with closely for the last 5 years or so, and to whom nowadays I'm doing almost everything in the therapeutic realm I do in close partnership with her, we made the conscious decision several years ago that the best way we could approach this was to try to address the needs of any patient who presented themselves to us. that we were going to try to meet each patient where they were and whatever variant they have, whether it's a single letter misspelling or a larger change in the DNA. We're going to try to do our best for them with whatever technology was available. And that's how we ended up helping the baby widely known now as KJ. He was born at the right time when we were sort of ready to go. He had a condition, extremely rare CPS1 deficiency. He had a misspelling that was unique to him effectively, or unique to his family. But we had consciously geared up to the idea, whoever shows up, that's the person we're going to try to help. Doesn't matter whether it's an N of 1, doesn't matter if it was an N of 100, we're going to try to meet them where they were. And so we made an all-out effort to help him, and it was successful. We were able to make an editing solution that corrected his misspelling. We were able to deploy it using existing technology, and that was important. There was existing technology we could draw on. It wasn't something where we were trying to make an entirely new therapy from scratch. We were adapting a therapy that already existed and had been deployed in adults, and we just redeployed it for an infant, and then used the nature of gene editing, the programmability of CRISPR, where you're basically tweaking one component that's like a GPS, and instead of pointing to one gene, you point it to another gene. We pointed it to KJ's misspelling. And to all appearances, the drug was effective in that it made a very, very devastating, life-threatening disease into a much milder form of disease. And he's doing quite well. And that's the approach we consciously took in order to try to be more equitable in how we're developing these therapies. Now, can you scale that up? That's an entirely different question. You can't mount an individual, all hands on deck campaign for every single patient who comes before you. We need to think about scaling up, and we need to think about the economic viability of scaling up and being able to deploy these to every patient, whether they have a common variant that's shared by many, many, many people, or whether it's someone who has a variant that's unique to them. And regardless of what disease they have, whether it's a more common rare disease or whether it's an ultra rare or nano rare, or even an N1 variant.
Right. And I mean,
we will, sorry, I was just going to say, we will return to the cost concept in a few minutes, but Olivia, would you like to add anything?
Yeah, I mean, I agree that it's important to try to make if the scientific, it's plausible, it's a plausible mechanism, so it should be approached in that sense, whether it's for one or multiple. And that's where we're seeing that the N of 1s that have been in trial that we're talking about, and whether it's gene editing or mRNA therapeutics, and especially in the neurodevelopmental space that I'm in, with a lot of children with different presentations similar, even though the etiology may be different. There's hundreds of known genes that are presenting so similarly like that. And so that's where the proof of concept and the improvements that we see and change in disease course that we're seeing even can be applicable to many more beyond the single one patient.
Thank you both. Let's take a step back and kind of start at the beginning. Let's pretend somebody gets a diagnosis today. How do they go about initiating an N of 1 trial? What does that journey look like?
That's a very big question.
I know.
But I would say starting with understanding the genetic variant and what's the presentation. Does it present with more of a gain or a loss mechanism? That's going to be important to understand, plus functionally characterizing it if needed. On top of the design, then followed by the design of actual therapeutic, whichever way that goes. And that's engaging just the beginning of it. Beyond also at the same time characterizing your natural history of yourself within disease, as you're trying to think about your symptoms now, how is it progressing, and how would that intervention change that potential course. I mean, I think that's just the beginnings of it, beyond going to actual production and then delivery, beyond IND approval at the regulatory level at the FDA, and then implementation in an institution that will support the delivery of it and have the resources to deliver it with expertise in a methodologic, rigorous manner, I think is important to maintain, because there's a lot of promise of N of 1s, but what does that mean? And, really making sure that it's delivered appropriately, I think is so important. It comes from root understanding of the variant.
Thank you. So it sounds a lot like in the beginning, rare disease journeys can be similar, which is what is it, what's affected, how does it work, how are other people affected, and then you kind of decide how many are there. And if it is a very small number or even just one, maybe that's when you decide we have to take this different approach versus the more common approach. Kiran, would you like to add anything to that question? Again, I know it's a
Yeah.
very large question.
Yeah, so it is a large question. So
It is.
to the point you just made about, trying to decide, oh, is it going to help more people? Is it going to help like fewer people? I don't think the approach necessarily needs to be different, right? The real obstacle here at the end of the day is delivery, delivery, delivery. Right?
Mhm.
So it's not just about whether the variant that you have can be addressed with an antisense oligonucleotide or with a gene editing approach, although that's an important consideration for sure. The fundamental challenge is what is the nature of the disease? What organs in the body does it affect to get an acceptable therapeutic response? Like maybe not a complete response, a complete reversal of disease, but to hit the important organs where you can make the most impact. You're limited by the technology that we have available to us, in early 2026 as things stand right now. Certain organs are easier to get drugs to than others. The liver, as it turns out, is much more straightforward for delivery because the liver's job in the body is to clean things out of the blood. So if you put pretty much anything in the blood, it will end up in the liver cells, unless you're trying really hard not to have it go into the liver cells. And so, I referred to baby KJ's case. What made that practical, eminently practical, is that we didn't have to invent a whole new delivery method. We didn't have to create a whole new type of drug. We were using already established technology, and I think that's probably like the biggest consideration as to whether it'll be a straightforward campaign or whether it'll be a much longer journey. In his case, we're using technology that scientists, that companies had been working on for many years, that I'd been working on in a totally different domain, heart disease. As I said, I'm an adult cardiologist, and so my patients are actually 70-year-old people who come in with heart attacks. It's the leading cause of death. So I spent quite a few years, and many people spend many years, working out the delivery technology to get gene editing tools, to get CRISPR into the liver. Once that was established, and once it was in clinical trials in adults, and there were signs of success, the path had already sort of been trodden. And so with KJ, we didn't have to create something new out of whole cloth. We could actually go to an existing delivery approach and adapt it, make it fit for purpose. Sure, KJ was the first infant to ever receive it, but fundamentally it was the same drug. Because of the programmable nature of CRISPR, all we had to do was tweak the GPS component, and instead of pointing to a heart disease gene, now we're pointing it at his misspelling. But for all the credit that Becca and I have gotten for what we've done, like conceptually on paper, it was actually very simple. It was just change the GPS and then make the same drug and then give it to the kid. And it seems to have worked. And so I think we're approaching an era where if you have a disease that is centered in the liver, and if you can make a change in the liver, then I think we have ways of addressing that. And in my mind, what flavor of disease and what gene is involved and what misspelling is involved, how common versus rare versus N of 1 it is, isn't so important, because the nature of the CRISPR gene editing, the technologies, it's just changing the GPS. And it's just as easy to change the GPS for an N of 1 mutation variant that one person in front of you has, versus changing it to make a change that would potentially help many, 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 many people. Technically, it's the same exercise. So then it just becomes a question of how, can you manufacture those therapies at scale? The other consideration is regulatory, like how amenable will regulatory agencies like the Food and Drug Administration be to you changing the GPS and then using that drug, the same, basic drug, but just, fine tuning it for each individual person, how agreeable they will for you to do that and just do it over and over again without having to do a lot of work each time you change the GPS. 3 years ago, I was less optimistic. It really seemed like the FDA and other regulatory agencies were going to say, well, if you're just changing the GPS, that's still a new drug and you have to do all this testing. You have to do all these animal studies. Got to really show us that this is like very safe and effective and whatnot. But over the last 3 years, we've been working very closely with the FDA. I've had something like 6 or 7 meetings with them over the last 2 years. And together, we've taken that journey. We've gotten to the point where, at least with this gene editing technology, with CRISPR, gotten to the point where we realize, it is the same drug over and over again. If you're just changing the GPS, it's the same drug. You don't have to do all these studies all over again. You can just make the drug and basically deploy it in the next patient. And so we've made a lot of progress, and I think that's opening up some very exciting opportunities. Now, does that help the vast majority of patients? Unfortunately, not yet, because only a small minority of patients have those diseases where there's something that needs to be fixed in the liver, which we can do now, versus in other parts of the body where we don't quite yet have the technology to deliver the CRISPR technology. But we're getting there. Lots of people, not me personally, but many of my colleagues all around the country, all around the world, are working very hard, very assiduously to try to make these delivery technologies to go to all the different organs, like the heart, which I care about since I'm a cardiologist, the muscles, the lungs, the blood stem cells, very importantly, the brain, the central nervous system. It will happen in time, but we are not there yet.
We've had a lot of questions just come in as a result of your reply, and this is great.
Oh boy.
Because there's some good follow-ons.
Okay.
I'll just go through them in order because I think it might be hard to sort of put them in perspective. So the first one is, how much does an N of 1 trial cost? And maybe you can talk about how much does it cost before you get to the clinic and how much does it cost when you're at the clinic?
So my perspective on that, so it's all about the context, right? So for the first time you do anything, it's going to be much more expensive. As I alluded, we were in a situation a few years ago where if you did anything at all, the FDA or others were going to consider it to be the first time. And so it was going to be very expensive to do it each time, even if you were doing more or less the same thing over and over again. We're now at the point where if it's considered the same drug, the first time you do it, it's still going to be expensive. It's going to be, like several $1 million or whatnot. But then the next time you do it, it's going to be much less expensive because you don't have to do things over and over again. You don't have to do lots of animal studies. You don't have to do this, that, and the other thing. So then it becomes much less expensive, and then you do it again and again, and it should iteratively, the cost should come down. And then if you can really get to the point where you're scaling up for a clinical trial, there are going to be clinical trial costs. You can't avoid those. The reason those are expensive is you have to do things very meticulously, very rigorously. You have to collect data. You have to do everything in exactly the right way to satisfy the FDA that, nothing but the most rigorous, high quality information is being collected and being relayed to the FDA so they can make the best possible decision as to whether a drug is safe and effective, which is those are the primary criteria as to whether they will approve the drug. So the clinical trial cost, separate from actually making the drug, that's going to be pretty substantial, pretty daunting. But if you can get to the point where you can get an approval from the FDA or another regulatory agency, depending on your jurisdiction, once it is approved, then you're no longer talking about clinical trial costs. You're talking about actually making the drug and delivering the drug. And you're talking about the potential for reimbursement from insurers, from payers, whether it's Medicaid or whether it's, private insurers. If you have the approval, then you have a fighting chance of being able to convince them, look, the benefit of this drug outweighs the costs of the drug. This is going to have a transformational effect for a patient's health. It's going to save healthcare costs that otherwise are going to be incurred because they are sick individuals. If you can present the argument that the drug, whatever it costs, whether it's millions, whether it's hundreds of thousands, whether, hopefully eventually tens of thousands or even lower, whatever that cost that's being borne by the payer, if you can construct the argument that, look, it is worth it in terms of saving healthcare costs that otherwise would be incurred, and for the overall benefit of the patient, their well-being, the quality of life, their longevity, like those have to be factored into the equation as well. If you can successfully make that argument, then you potentially make this economically viable, right? Then you can scale up and you can get these types of treatments to all the patients who need them.
Thanks, Kiran. Olivia, you're in a different part of the body, in the CNS and other similar areas. Do you see a similar amount of cost, or do you think it will be different depending on where folks need to be focused?
I mean, I think I have to agree with Kiran. I mean, I'm dealing with babies and children with seizures, often day of life 1, and significant delays where they may have failed, 10 daily medications every day, and they're still seizing away, and they're not able to walk or talk. And these are things that are incredibly severe presentations of disease that any level of intervention could be improvement in quality of life and symptoms for the actual well-being. Many of these diseases also in childhood neurological disorders present with increased risk of death, and these are comorbidities that are beyond, just the day-to-day seizures. And so there's an enormous socioeconomic impact, not only on the patients within family, but, their parents' employment. There's lots of across the board examples of how this level of intervention can improve. I mean, we've been seeing that in N of 1 ASO trials that I've been doing also for several years at this point, and seeing dramatic change in development that is beyond what you would normally think. And for example, for one patient with SCN2A related disorder, and that's in collaboration with NLORM Foundation, where a patient started walking at 14 years of age, and he was previously referred for hospice at 2 years old. And so that just makes you think of what's the boundaries of developmental change, where's our synaptic plasticity, what's the limits of intervention. But I think that these are really important questions that come up from learnings from the trials that we're doing. But to Kiran's point is that this does come with a level of rigor that is really important to maintain so that we can show this evidence and then make it more accessible as we keep implementing these trials. And there's a change. I do think that, as Kiran mentioned, there's been a shift, I believe, in regulatory as well on the level of FDA. And we're just, I think, much more collaborative at this point because we all want to bring this kind of opportunity forward for all of these children and adults and elderly who probably have many undiagnosed disorders that, could improve from a targeted intervention. And so I think this is just changing how we think about healthcare and what's possible.
You've both mentioned rigor and safety quite a few times, and I know that that's more than just each of you, taking this on. How many people does it actually take to successfully initiate and run an N of 1 trial?
A village, I would say, because I mean, that's an enormous community that brings together a single patient. And I mean, I would say when you think about dosing day, that's only one aspect of like a huge effort that comes from an enormous group of scientists across the board, preclinical to all the way to like the nurses or whoever research coordinators that are delivering it. So I think it's very difficult to say numbers of people, but it's usually a huge team.
Yeah, I would say in baby KJ's case, we tried to do the math, and it's challenging because a lot of the effort, a lot of the expense was manufacturing of the drug, which happens at companies, and they have staff, they have teams who are making the different components of the drug and then bringing it all together, and you have to fly it from one place to another place, and then you have to get quality checks, and that happens yet another place, and so a lot of people end up touching in one way or the other. But we did the calculation, it ended up being on the order of 200 to 300 people who we think had pretty significant roles in making the drug. Now, again, it's not 200, 300 full-time jobs, right? It's different people who at different points in the process, whether it's the manufacturing, whether it's the drug design, whether it's the regulators, whether it's the clinicians who are taking care of the patient and who are there on dosing day, as Olivia mentioned, and who are giving the follow-up care, right? A lot of people end up becoming involved. And I should also say that, it depends on whether it's gene editing versus an antisense oligonucleotide versus something else. But at least in my world, gene editing, at least up until now, these sorts of exercises, these N of 1 sort of efforts have not really been research per se. They've been more use of an experimental drug as part of clinical care. Like there's a desperate need on behalf of the patient. And so you mount an all-out effort to try to make therapy for that patient. And then you give it to the patient. And then the context is really clinical care. It's, what they're receiving in the hospital to treat their disease. And I think, again, that speaks to the larger issue where at least what I've been involved with gene editing, up until now, it hasn't really been part of a clinical trial. And so that's okay, but at the end of the day, that doesn't get the football closer to the goal line, which ultimately is getting these sorts of drugs approved, right? If you're just doing, hey, there's a patient who needs this drug and we make the drug and we give it to them, and we're not doing it in the context of a clinical trial. We're not gathering the data,
Yeah.
on safety, on efficacy. We're not doing things in a very, very rigorous way, very expensive way, but a very, very rigorous way. And then have those data that we can give to the FDA and say, hey, look, we have very strong evidence. for this individual and this individual and this individual, and as much evidence as is needed to make a strong case. If you're just doing, these isolated, let's call them N of one exercises that are more clinical in nature than research in nature, you're not going to be collecting those data, and then you're just doing a bunch of one-offs. I don't like to put it quite those terms, but that's effectively what it comes down to, and doesn't
Yeah, could you elaborate?
onto the goal line, as I said.
Could you elaborate
That's
on that a little bit? Because there's been some. Oh, sorry. Go ahead, Olivia.
I was going to say that's a very important point too, that I 100% agree. It's very important to distinguish like just implementing a change in an individual in your clinical care in a personalized approach maybe, but not actual with rigor necessarily on the level of regulatory. That's different in research level. And I think that is really important, as Kiran mentioned, to move the needle forward beyond just individual level care. Yeah.
As an extension of that, are there any examples yet of the FDA using N of one data to approve a drug, or accepting N of one data as part of a larger package to continue testing the drug?
Not that so there are different levels of data. And so what they want to see first and foremost is like unambiguous clinical trial data that support safety and efficacy. That needs to be the core of the package that's ultimately submitted to the FDA, the application to actually get approval for a drug. That is not to say that N of one sort of exercise is more clinical exercise, that data from those can't, that's not to say that those data aren't useful and can't support the application, but it's hard for me to envision, and I'm not aware of any example where those sorts of isolated data from almost these isolated cases together can be aggregated and make a strong enough package on their own to support approval. I do think you need a formal clinical trial in place to be able to get that approval.
Yeah. Although I would say in the developmental and epileptic encephalopathy space, there's been cases of N of ones that have been in other countries as well that begin and then have evolved into clinical trials that are bigger clinical trials, not necessarily approved yet, but they're in clinical trial that started from an N of one for a single patient. So I think there is precedent in general for the regulatory potential. Although I'm not sure if it's all across the board approved, but there's a pathway there that has been traversed.
Yeah, the best case scenario is absolutely like you start with an N of one exercise and you see some signs of success, and that inspires, your own team, colleagues to commit more fully to that, and then launch the clinical trial in the fullness of time, and then be able to get to an approval. I think that's the ideal scenario for all of us.
Mm-hmm.
So I'm going to combine 2 or 3 questions into one here, because people are asking similar things, which is, maybe I find out my kid is 2 of 30, and, as a patient advocacy group, how do we make the decision that we do an N of one, try to get that data, and then push something else? Or how do we decide we're going to have something larger and treat all 30 at a time? And part of that, I think, comes down to how much does it really cost? If you have a very wealthy donor that says, I'm going to pay for everything, you kind of have to make these complicated decisions. I guess what I would ask, sorry to simplify it, is how much would it cost if one person says, I'm going to fund the whole thing? And then as an extension of that, how likely is it that doing that will be able to be applied to the broader community? The data, that is.
Yeah, I've had a complicated set of questions.
I know, they're all very complicated.
Yeah, I mean, what I would say is I can only tell you what we, Becca and myself, have decided to do. And part of it is because we want there to be as much equitable access to the types of therapies we're trying to make. We did baby KJ's case effectively as an N of one, for better or for worse. I think for better. I think it was important to do that. We needed to open the door. And then after that, we decided, in part because of feedback from the community, from patients, from patient advocates, from our fellow physicians and scientists, from the FDA, we made the decision that we weren't, at least for that set of diseases, which KJ had, one of those diseases, CPS1 deficiency, we made the decision that we were not going to do any more N of ones in that context. We were going to do everything we could to launch a full-scale clinical trial as quickly as possible. Because otherwise you end up in this situation. How are you deciding who's going to be the beneficiary of a N of 1 effort? And there are many people who have need. How do you pick number 2 or number 3 or number 4? And then if you're not working towards an approval and a viable economic model where post-approval you can actually get reimbursement, you're going to run out of resources, right? It doesn't matter how much wealth is being leveraged. You're going to run out of resources. And then whenever you run out of money, the party stops. And if you did 5 individuals, they got lucky, but everyone else is left outside, right? That doesn't feel great to me. So a clinical trial, at least you open, at least in principle, anyone has access to it, anyone from anywhere in the world. Now there are practical limitations, but at least there is the theoretical potential that anyone who has a disease has a fair shot at being enrolled in that trial and have the access. And hopefully you get to the approval, and then everyone in principle should have access.
Is it the same, Olivia, in neuroscience?
I mean, these are some very difficult questions that are being given to us, and I think there's no straightforward answer. I would say that, if it was, a lot of the DEEs as well, there's just so many hundreds of variants within a gene that can present differently as well. So this is a common question that comes up, like, are you going to go after one particular variant if there's more common recurrence of a single one versus other types of approaches as a design into a haplotype-based phasing, where you might not actually just target the mutation or the variant, the causal variant. If it's a different SNP, then maybe that particular same drug could be ASO or whatnot, could be used for other people within that community, even if they don't have the same exact variant. And so there's different ways of, I think, creatively trying to approach that challenge, because it's across the board. Everybody wants to promote equity and also advance science as fast as we can. And I do think that we're on, it's like a tsunami wave, I feel like, in the space of neurodevelopmental and epilepsy DEEs. But I think that it's not so straightforward of a question when you ask, who gets to go first. But at the same time, not everybody wants to go first in an early genetic therapy trial either. So I think there's lots and lots of nuances to that. Again, remembering that as we talk about this, it's beyond just the concept of it. There's a lot that comes into actually being a subject in a trial like this, for the patients and the families. These are, again, within clinical trials. So the visit windows are narrow. There's lots of things that have to happen on a level of rigor that's not, typical clinical engagement. And so I think that needs to be also something that comes into consideration as you think about whether you're ready for something like that right now.
So there have been some other questions that have come in sort of around how do you go about doing this? And what I mean by that is, the question is, how do I know if I should do it on my own? How do I know if I should go to a nonprofit? Or how do I know if I should go to a company such as Lorem that you mentioned, Kiran, that has started doing more of this? How do people decide the best way forward? I know, a very difficult question. There's no easy ones.
I don't know. Olivia, you may have a better sense of.
You're going to throw it to me, Kiran. I would say, there's incredible collaborators across the board. I think if there's existing precedent and there's been proof of concept somewhere, for example, with many of the NDDs in different advocacy groups, and Lorem being one of them, I think that's an outlet that we could you can leverage. I think that it's, again, comes down to what's the disease, because there's some approaches that make more sense. But I think across the board also, I would say that beyond just making sure that it's safe, that there's something that's actually potential for efficacy beyond just being delivered to you and safe without any response, because of course somebody could make something that doesn't have a side effect, but then there's no improvement either. So I think these are things that are really important in maintaining for kind of a vulnerable population that is looking to seek a treatment pathway forward. It's really important to look at resources that have expertise and credibility there too, I think is important.
Anything to add, Kiran?
Yeah, I would just say, again, I'm ultimately at the end of the day a very pragmatic person, and so speaking for myself and only for myself, um, I want to do what feel I can get done like now in this time frame, like what is in front of me, right? And for now, at least in the area I work with, gene editing, CRISPR, that means the liver. If there is a case that involves a gene, a misspelling in the liver, where making a change in the liver could potentially be helpful, I'm very open to hearing that. If it's beyond the scope of the liver, I don't feel we're quite there yet. Doesn't mean that it's not worth pushing on it and worth campaigning for it. It's just not something I personally am in a good position to help with, is what I would say, at least as of early 2026.
We've talked a lot about how these unique trials require a lot of thoughtfulness, right? Because you're trying to do things differently. Do you guys have good examples of how these different approaches have helped influence the FDA or other regulatory bodies to change how we're thinking about smart or bigger trials? But then also, are pharmaceutical companies looking at things differently as well? So maybe another way to put this is, are the learnings from N of 1 trials changing the way that the FDA, different companies, or different nonprofits are approaching drug development?
Well, I would say the FDA, with respect to my area, gene editing, CRISPR, so forth, it absolutely has had an impact. So just a couple weeks ago, the FDA announced and published a draft guidance on a new framework, let's call it. That's what they're calling it, a framework, not necessarily a pathway, but it's called the plausible mechanism framework. And there's no ambiguity about it. It was directly inspired by baby KJ's case. In the press conference, several leaders at the HHS and FDA explicitly said that this was inspired by KJ. And so I think there's a dramatic example where a single N of 1 case actually did make a pretty big impact. And that framework at least offers the possibility that we can streamline the clinical trial process to be able to make individualized, personalized genetic treatments. And there's openness there to both gene editing as well as antisense oligonucleotides if you actually look at the draft guidance. The devil's in the details, but at least at a high level, there does appear to be openness to doing relatively small phase 1-2 clinical trials with relatively small numbers of subjects in a non-randomized way, using appropriate comparators, endpoints. I know that's a very challenging thing to talk about nowadays. But assuming you can make a strong case that your biomarker, is a truly rigorous biomarker, potentially getting to an accelerated approval with as few as 5 to 10 subjects. If you can really show, look, this is safe and this is working really, really well, there appears to at least theoretically be openness to that in a way that we have not seen before. So I take that as an encouraging sign. Now, actually getting it done, we'll see. I mean, my group, we're very much intent to try to take advantage of this framework and get some approved therapies in the coming years. And, talk to me in a couple of years and we'll see where we stand and just how easy or hard it turned out to be.
Yeah, I agree. I mean, I think there's implications across the board as we're seeing things moving faster through and showing efficacy in the N of 1 state, that it is causing some ripples across regulatory, and that there's increased in acknowledgement of how these kind of interventions can have significant impact. And so we're also seeing that, for example, in the developmental and epileptic encephalopathies, where some of the industry trials are no longer required to have a sham placebo LP arm, and so these are things that where these children wouldn't be waiting with 6 months or whatnot, to see whether or not they get intervention when that they're progressing. And I think these are changes that are happening as well that impact bigger trials. Yeah.
So believe it or not, we are at time, or we have 1 minute left. Is there anything that you guys would like to leave the audience with, something to take away from this discussion? Olivia, would you like to go first?
Sure. I think that, I appreciate this particular format because it is about empowering patients to really understand what is the current landscape and what's possible. And I do think that there's an incredible amount of exciting science happening that is making waves forward for potential therapeutics that are, have already caused a disease modifying effect. And so hopefully we can engage together because it's a big collaboration between academia, industry, and of course, families and patients advocacy groups.
And I would just close by saying, it's still very early days. We have some promising early signs of success, but there's a lot of work to be done, and we will not
Mhm.
get that work done unless we have all hands on deck, all of us working together to push this forward.
Right.
Thank you. I want to thank our panelists, Dr. Musunuru and Dr. Kim-McManus, for their perspectives and roles in helping us understand and get access to these sorts of treatments.